Because the liver can only produce so much of the natural alpha-1 microglobulin, basically what you need is a superabundance of alpha-1 microglobulin, or ROS-guard as we call it, to counteract the superabundance of the reactive oxygen species of all kinds.
Hello, my name is Mike Ward. I'm the head of content at Scrip and the Pink Sheet. We're here in Berlin at the Bio-Europe meeting, where pharmaceutical companies, biotech companies and investors all get together, take the finger on the pulse of what's actually happening in the industry, maybe even actually engage in discussions, whether it's for investments or partnering deals. One of the areas which is a hot topic and has been a hot topic for a number of years is the area of oncology, cancer treatment. While a lot of the talk has been around immuno-oncology, actually there are still sort of opportunities for sort of the old classic ways of treating cancer. One of those, of course, is radiotherapy. But of course, one of the sort of the challenges around radiotherapy is the side effects, the damage that is caused to both sort of kidneys and bone marrow, and the fact that actually you have to restrict the amount of dosage you can give, has in the past sort of held it back as a way to treat cancer. However, a company called A1M Pharma has come up with an approach that actually looks like it is protective for those against those effects. I'm joined by Martin Austin, who is the chairman of the board of directors at A1M Pharma, and he's also heads up their business development effort as well. So Martin, nice seeing you again.
Hi Mike, how are you doing?
I'm doing fine. So tell us, this compound you've got, what is it, and what evidence have you got that it has some sort of protective effect?
Absolutely. I say a compound, it's actually a protein, and a protein which in its natural sense is found in all vertebrates. So everything that's got a backbone has an A1M. Yeah. A1M is alpha-1 microglobulin, which is a small protein which is secreted by the liver when you are under what we call oxidative stress. Right. So anything where a molecule's been pulled in half and one half is positive and the other one's negative, the positive ones will always try and find a home, and that home is usually on a membrane. That membrane starts to misbehave and suddenly things are going wrong. The place where it happens most frequently is of course at the kidney, because everything that floats into the blood is taken to the kidneys for filtration, and if those filtration membranes start to go wrong, then you're going to end up with acute kidney injury. So the reactive oxygen species are at the root of the trouble. Many diseases create these reactive oxygen species, and as a result, they are one of the worst bad guys you can get, because they will ruin your kidneys. However, as you pointed out, radiotherapy is actually a controlled use of reactive oxygen species to try and kill cancer cells. Yeah. So what happens in the cancer cell is that as it's trying to divide, its DNA, it separates. If you remember, everybody's seen this one in school, that the DNA, the chromosomes pull apart. When that's all exposed, the radiation knocks strands apart within the DNA, and when that happens, the cell biology down at the cytosol level will say, oh, this is a cell which is going badly wrong, and apoptosis, the programmed cell death, starts to take place, and that's what you want to have happen in the tumour. Wonderful, except for the fact that the energetic particles that you've created are going to go down to your kidneys, and they're also, in the radiation event, going to start affecting your bone marrow at a certain level. Now, what we found with alpha-1 microglobulin, ROS-guard, our product, ROS, reactive oxygen species guard, that's what we're actually making in a recombinant form, so that because the liver can only produce so much of the natural alpha-1 microglobulin, basically what you need is a superabundance of alpha-1 microglobulin, or ROS-guard as we call it, to counteract the superabundance of the reactive oxygen species of all kinds. So we're able to deal with heme particles from hemoglobin, we can deal with hydroxyl radicals, we can do anything you like, including the ions which are created which are used in what's called peptide receptor radionuclide therapy. It's a bit of a mouthful, but PRRT is becoming very, very, I won't say popular, but very attractive to pharmaceutical companies, because it's a means of being able to inject radioactivity into people in a way which is then targeted, not exclusively, but goes to and sticks to the tumours which are metastasized are all around the body, so you can't get to them surgically. The cytotoxics will kill everything that's around there, so you want to try and get something which will target that area. So somatostatin has been used as the longest used molecule. Clearly it was started out used in acromegaly. Acromegaly is actually due to a tumour which is the same as the ones which we are looking to treat, the neuroendocrine tumours. It's the same tumour type. So there's a whole paradigm which has grown up around PRRT. The trouble is, if you inject 5 mils, 2 mils, or whatever the radioactivity is, only so much of it will reach the tumours. The remainder of it goes to your kidneys, and is also absorbed into the bones. So if you had something which naturally will neutralize the charge on those energetic particles, then the radioactivity per se itself will not really matter. And you say, well, how could that be? It's still radioactive. So it is. But the radioactivity creates ions which do the damage. Ions are reactive oxygen species. So if, as they're created, we eat them, effectively electro-neutralize them, which is what that protein does in its natural form, then basically for the half-life, and I don't mean the radioactive half-life, I mean the pharmaceutical half-life of the radioactive substance, can pass, and it'll be eliminated without damaging the kidney. And what we've also shown is that Rosgard protects the stem cells in the bone marrow, which has been the thing which has the greatest effect on patients, because as the bone marrow dies, you either have to have, as is done in some cases for people receiving radiotherapy, they have to have a bone marrow transplant in order to restart the blood generation. We can stop it at source by preventing the radiation from killing the cells.
So Rosgard, where was it sort of discovered or developed first, and what was the original plan for the molecule?
Originally, well, the molecule was actually handed to one of our founders many years ago as his PhD thesis, about 40 years ago. He's gone on and characterized it, found out what it does, how it works, where it works, and has done all these things. When we first started the company, which was in 2008, the thought was, because he teamed up with an obstetrician and gynecologist, the thought was that this would be ideal as a safe means to try and protect mothers from the hemoglobin, which leaks through the placenta of a baby, or from the baby through the placenta into the maternal blood circulation, where that hemoglobin breaks down into heme. The heme affects her kidneys, and she suffers a nephropathy, which is associated with what we call preeclampsia. That's quite a complex disease, but the end result is that the kidneys are being attacked. That remains true, but clearly it's a pretty distant target by comparison with what we're talking about with radionuclide therapy. But the principle, the platform we're talking on, is that we neutralize reactive oxygen species. So we can be used in either case, and one is actually going to be, the underlying principle treats all those diseases, which is great.
So in your development, where are you at the moment?
We are at the point of submitting for a clinical trial in PRRT as to establish a proof of concept of the mechanism of action and safety in patients. That will then open the door to all the other indications where reactive oxygen species are the criminal in the investigation, and will open up opportunities for us to partner with companies that need a suitable way of being able to neutralize those particles and protect patients after all.
I'm presuming this will be attractive because radiotherapy is going to be less expensive than some of the immuno-oncology compounds that are being created.
I have to say that the pricing of what we've produced would probably not get up to those levels, because if you can from the basis of the clinical studies that I talked about, our development costs have not been that high. So whilst others have spent hundreds of millions, we haven't. We've spent a lot less in order to get to this ability to produce a proof of concept in the clinic.
So this is a partnering meeting. No doubt you've been gone out sort of broadcasting, what you have shown. What sort of interest is there in radiotherapy?
As it's such a novel idea, I have to tell you that the results for the protection of the bone marrow only were confirmed in the last couple of weeks, so we haven't been able to tell too many people about it. We made our press release this morning to say that we filed the patent for that. So this is all very novel, and people are going, but nobody can do that. It's never been done before. The intrigue is very high. The interest in being able to enter this opening area of peptide receptor radionuclide therapy, which has just attracted a deal at the 4 billion level in Europe, and is coming up for regulatory approval as a mechanism of treating cancer in the United States. So everybody's very, very interested in that. Everybody we've spoken to so far at this conference has said, wow, in one word.
So you can expect lots and lots of acceptances to invitations at the next BioEurope Spring meeting?
That is a shoe-in. I'm coming. I'm definitely going to be here. Okay. That's if somebody hasn't already given us an exclusivity deal which says that we're tied up. Not that I would turn it down, but we're coming to BioEurope Spring anyway.
Yeah. Okay. Cheers, Martin. Thanks very much. Bye.